All right, I guess we're going to go ahead and get started. I hope everyone had a good lunch and got to see some posters. I'm delighted to be moderating this panel. From my perspective, really, in science and medicine, the immune system is the only system that matters. And so we have a wonderful panel of experts on immunology. And so I'll just briefly introduce them, and they will give a short overview. Immediately to my right is Dr. Mark Soloski, and Mark is a professor of medicine at Johns Hopkins. He's really been a pioneer in research on the immune response and trying to figure out what is happening initially from the molecular and genetic standpoint in both the innate and the adaptive immune response to Lyme. Next to Mark is Dr. Bill Robinson. Bill and I actually go back a long way. So Bill is in the Department of Rheumatology at Stanford. He's an MD, PhD. I actually helped Bill start his lab and get going. We were unpacking boxes many years ago, so Bill and I go back a long way. Next to Bill is Dr. Monica Embers. She, as she mentioned earlier, works in non-human primate models, and so has been an expert in trying to figure out what is the similarities between the primate response, immune response, how to develop, understand the antibody response to develop better diagnostics and potentially guide cures. Next to Monica is Dr. Nicole Baumgarth, and Nicole is at UC Davis. She has a really interesting perspective coming from the coevolution of looking at the immune response in mice and what can we learn from how the immune response has been built up in mice and how Borrelia actually can potentially co-opt and evade the immune system in humans. And then at the far end is Dr. John Aucott. He's a professor of medicine at Johns Hopkins. As he said, he was adopted by the Department of Rheumatology. He's cut his teeth in infectious disease, and I guess rheumatology took pity on him and brought him into the fold. And so, but he has been a champion of Lyme for many years, being the director of the Lyme Center down in Hopkins, and he's actually going to kick off our panel today.
Thanks. I thought it was appropriate actually that Monica's a non-human primate translational researcher. Nicole's a mouse translational researcher, and I'm a human translational researcher. So that kind of gives you the spectrum of the, the different hosts. So I'm going to start today, and I'm really going to emphasize subgroups and what we've done in the SLICE study to identify subgroups. So I'm going to talk about three areas of subgroups: the clinical manifestations of Lyme disease, the microbiologic, and then the immunologic, which are the three subgroups we study. So if you look at these three slides as a clinician, you may be asking, how do we diagnose Lyme disease? Who of these three people has Lyme disease? And so let's look at some subgroups. So the first person actually is the classic, doesn't really look like Lyme disease to the average doc because it's got that big blister, but it has a positive serology and a positive skin biopsy for Borrelia DNA. So we get that one. The next subgroup actually that we've identified with work with Mark Eshu at Ibis Abbott are people that are seronegative but skin biopsy positive. So here's a subgroup defined by the microbial PCR findings and the rash, but is seronegative. And then a recent group we've actually found that are both seronegative and skin biopsy negative for Borrelia. So what do these people even have? To a clinician, you look at that, it has central clearing, it's a ring, you think it's Lyme disease, but by all our criteria, this is a subgroup that we can't demonstrate Borrelia in or antibodies in. So subgroups are important. We think it's the key thing to defining the initial diagnosis of Lyme disease. But as a clinician, it's even more important to measuring the outcomes of therapy, because what I'm interested in as a clinical researcher is which of my patients are going to get better. And again, we think it all comes down to subgroups as defined by their clinical phenotypes, their immune response, and then the original Borrelia that infected them. So how do we figure this out? So we started the SLICE study at Hopkins about 8 years ago, and this is a clinical translational research program where patients are diagnosed with erythema migrans rash, and then the first box in the upper left is in our clinical labs, we define all their clinical manifestations, their neurologic data, cognitive testing. We get all that metadata that our collaborators want to know about who are these patients with Lyme disease. They all have an erythema migrans rash, and they get skin biopsies. And so the skin biopsy tells us whether they have Borrelia infection, as I just showed you. This work's done with Mark Eshu's group at IBIS, and we're able to microbially define these patients that they have PCR positive Borrelia identification of nucleic acid in the skin biopsy specimens. So this is one half of what Monica said. It's the pathogen on one side, and on the other side is the antibody response. And this is the immune profiling we're doing with Monica Embers and with the group at Stanford led by Bill Robinson, that we're looking at the host immune response to these Borrelia infected patients. And then we've completed out with transcriptome work and metabolomics work. And actually, I didn't have enough arrows for the slide, but currently I have about 20 collaborators that take advantage of the SLY study and these well-characterized patients. And this has resulted in a lot of publications, actually, in terms of PCR identification for early diagnosis, tests that have been developed with Mark Eshu, immune work with Monica Embers and Bill Robinson. And we're getting the knowledge advanced through one-to-one collaborations, but we really are grateful to the Cohen Foundation for allowing us to link this into the next stage, which has been called the LymeMIND project. And basically it's taking all of these arrows and taking this data into one network of big data analysis that will occur here at Mount Sinai. And why this is so important is because we've made one-to-one collaborations, but this is going to allow us to have this data now analyzed and looking for connections between our collaborators and their different projects through the LymeMIND project. So we're very excited about this. We're also excited because there's no lag time for this project. Because we've been up in business for 8 years, we're using pre-existing samples and data that's already been collected to feed into the LymeMIND project. So it's actually already started with analysis of samples that are previously collected. I was asked to end with one, what my dream would be kind of question. And so that would be, it's been hinted at before, but all this work is being done on blood. But actually, as a clinician, we need to be able to do this work on tissue, surgical pathology specimens. And now that the Lyme Center at Hopkins is open, we're seeing more and more patients that have biopsies of neurologic tissue and joints. And right now, we need to be able, I think, to expand this kind of analysis to tissue-based diagnostics, because that's really lacking in the clinical realm, is understanding how to analyze tissue other than blood samples.
Right. So our angle is a little different, as you heard. We are really considering Lyme and Borrelia burgdorferi in its life cycle, and one of its natural hosts and one of its most important natural hosts is the mouse. And when you think about that, it needs, Borrelia needs to establish persistence in this animal, then I think you need to then you're also thinking about it for just a few seconds, you come to the realization that the only way that a bacteria can establish persistence in a host is that it somehow subverts or outmaneuvers the immune system. So I'm an immunologist, and we are very interested in understanding how Borrelia burgdorferi establish a persistence in an immunocompetent host. We don't do that because we're interested in curing mice, of course. We are interested in curing humans. But the mouse and the human have immune systems that are actually very, very similar. They have differences, but they're also very similar. And I think the principle that I outlined in this slide is really what is driving our viewpoint of how we see disease, infection, and also disease. If Borrelia over evolutionary time has established sort of a truce with the mouse immune system, in which the host, namely the mouse, allows the Borrelia to establish persistence without causing disease. That is good for both the Borrelia, because it establishes persistence, can fulfill its life cycle, but it's also good for the mouse, because the mouse is not diseased, and therefore will not be eaten by any of all these predators that are around. And so what we are thinking is that the mouse then provides this perfect balance. And I'm sure that there are humans that are infected with Borrelia that behave just like the mouse in terms of their immune response to it. But there are probably also humans, and we know about humans, that respond very vigorously with strong inflammatory disease. And that, of course, is because the human, as you've already heard, is not a natural host for the infection. It's a dead-end host. And so there's no evolutionary adaptation that has occurred. And so you have the full spectrum from a broad inflammatory disease in response to Borrelia, as well as one where you probably have very little inflammation, but you may have persistence. And so so what we are looking at then is what parts of the immune system Borrelia might have used to subvert, because those are the parts of the immune system that are probably most important for the Borrelia to switch off. Now, once we know what parts of the immune system are most important for Borrelia to switch off, then we might know what are the parts of the immune system that when we stimulate them can maybe help us to clear the infection by enhancing the immune response, rather than, as Dr. Lewis pointed out this morning, a sort of drug intervention. So if I could have the next slide. Oh, no, I didn't do the next slide, I guess. On this slide, what I'm showing you here is just, I want to show you two data that I think make the point that what I was trying to make. When you take a Borrelia-infected mouse, and you vaccinate that mouse with an influenza vaccine, what you will find is, or if you take a normal mouse, it develops a normal immune response. If you take a mouse that is Borrelia infected, it will not make an effective immune response, not only to Borrelia, but also to the flu antigen. Okay, can I have the mouse? This mouse, please, the other mouse. So here, what we have done is we have infected mice. We took the serum or the antibodies that were made in this mouse. If the mouse is not infected and you give this serum or these antibodies to another mouse, the mice are completely protected from a high dose of flu infection. They don't lose any weight, they're perfectly healthy. Now you take the serum from a mouse that is vaccinated while it had Borrelia infection, and now you can see that the serum from these mice are no longer able to protect the host from flu infection. What this shows you is that the immune response during Borrelia infection is altered in a way that the antibodies that are made are no longer fully protective. And this is not because the mouse is sick and just can't do anything. We have plenty of examples where parts of the immune response behave completely normal. The second set of data that I'm very excited about is this one. So what you see here is mice that are infected with Borrelia, and we are basically taking a little blood sample and we're culturing, you're asking how much bacteria, or are there bacteria in the blood, yes or no. Groups of mice. So if you take an immunocompromised mouse, a SCID mouse here, and you infect it, you will find that by about day 10, all of the mice, 100% of the mice that are infected have Borrelia in their blood. And these mice cannot make antibodies, they don't have cellular immunity, T-cell dependent immunity. So you will find Borrelia in the blood. But if you take a normal mouse, a normal immune system, you will find a tiny fraction of the mice actually have Borrelia, although they're all infected and they have Borrelia in their tissues. And it is, so bacteremia is not commonly seen even in normal mice that remain infected. And in fact, after day 10, you don't really see much in the blood at all. So now what we have is, and it doesn't really matter, we have a genetically altered mouse. And this genetic alteration doesn't allow it to make one particular type of antibody. And when we look in this mouse now, what we find is that the antibodies are very good at protecting from this bacteremia. And what I want to show you is, however, if we take the serum from these mice, the antibodies that are there, the IgG antibodies, and transfer them in the same way we are doing here, we cannot, in this genetic alteration, we actually have better protection. So we have a system where we have now demonstrated that with a small genetic alteration, by switching off one of the antibody responses the mouse is forced to make after Borrelia infection, we're making other types of antibodies that are higher protective. This is a second example that the immune response is not as effective as it would be, and that it's Borrelia that is actually changing the immune system. And so, again, we are excited that although this is done in the mouse, and this today is really all about human responses, we are grateful for support from the Cohen Foundation and from GLA, because we believe that these fundamental studies can really also help us understand why humans have so many problems with this infection.
All right, thank you. So my lab has spent a lot of time studying antibiotic efficacy, and we're left with a number of open questions about PTLDS. This is post-treatment Lyme disease syndrome, not post-traumatic stress disorder. So we found in monkeys that after antibiotic treatment, we were able to recover intact spirochetes using a technique called xenodiagnosis, where we place uninfected ticks on the animal, and they're able to send out chemical signals and take up the spirochetes. And so now what we want to know is, what is the phenotype of these spirochetes? Are they viable? Are they attenuated? Are they dormant? Do they persist in the long term, or are they eventually cleared? And are there indications of biological cure? So we know that Lyme disease is an inflammatory disease. And if there are remnants of the spirochetes, even attenuated spirochetes, they could continue to cause the inflammation that possibly results in the myriad of symptoms, chronic symptoms, that contribute to post-treatment Lyme disease syndrome. So one of our, what I see as a top priority for Lyme disease research, especially in this field of understanding post-treatment Lyme disease, is to identify the cause of post-treatment Lyme disease, and for the individual to identify that cause on an individual basis to guide proper treatment. You can imagine that if a person has a persistent infection, you don't want to treat them with steroids, for example. And we know that persistent infection, at least from the animal models, is a viable possibility. So we need to be able to identify treatment options that are effective. So like I said, we work in the rhesus macaque model of Lyme disease, and they, as I mentioned before, they're unique in that they have, this animal model allows us to control the infection, control the spirochete that we put in, but it's also highly variable compared to the mouse model. In fact, I've had some people tell me that the macaque genome is actually a little more complex than the human genome, or diversity, genetic diversity. So we know that rhesus macaques mimic the same signs and symptoms of human Lyme disease. Mice, for example, don't get a rash, they don't have infection of the nervous system, and we know that disease hallmarks such as the erythema migrans, carditis, arthritis, neuropathy of both the peripheral and central nervous systems, these are all observed in infected rhesus macaques. We also know that the spirochete burden in tissues is very small compared to what you might find in mice, as mice can serve as a reservoir host, or do serve as a reservoir host. So this is a blessing and a curse for me, because it's extremely difficult to find the spirochetes in the infected animal, but it also replicates human infection. So we've done studies in rhesus macaques, and what we've observed is the very variable manifestations that you see in human Lyme disease. In this particular set of experiments, which was 10 monkeys, we found erythema migrans rash that would be considered a bona fide EM in one of our 10 monkeys. And the other animals had some diffuse erythema. Granted, we put more than one tick on each monkey. And we also saw changes in serology. So what you will see here on these two bottom figures is the immune response. These are the antibody responses over time to two different Borrelia antigens. Those that were treated with antibiotics are shown in red, and those that were not treated are shown in blue. So on the bottom panel is C6. So we would expect that those treated with antibiotics would have a drop in their C6 titer, as many did, and even some animals that were not treated had a drop. And then if you look at the response to this other antigen, which is, I believe it's OSP-C, you'll see the responses come up and then drop for some animals and fluctuate a bit for other animals. We also had 2 animals that were seronegative by these standards. One of those animals had no pathology and no indication of infection, and we think it may have cleared. And then we have another animal that had, by xenodiagnosis, a persistent infection. So we're seeing these, like Dr. Aucott showed you, these different categories. We also see moderate pathology in a broad range of tissues. So here you're seeing lymphadenopathy in the axillary lymph node. This is hypercellularity about 11 months after infection in this animal. This is inflammation around one of the peripheral nerves. We think that this might be contributing to the symptoms, the variable symptoms of post-treatment Lyme disease syndrome. And then finally, we see rare persistent spirochetes in tissues that have pathology. So we looked in, this is the meninges and spinal cord of 2 monkeys, or this is the heart and spinal cord of 2 monkeys, and you see the small green dot. This is a antibody specific for Borrelia. So we believe that this is a very good model to look for, to test therapeutics, to understand persistence, and to look for biomarkers of biological cure. Thank you.
So my laboratory at Stanford is focused on studying the immune response and specifically the role that B and T cells are playing in effective immune responses. And in Lyme disease, we're very interested in defining immune responses that mediate successful clearance of the spirochete and thus are protective, as well as immune responses that result in post-treatment Lyme disease syndrome and thus are effectively pathogenic to the host. And we believe that really defining rigorously at the single cell level that the key B and T cells and their effector functions that are mediating the development of post-Lyme disease syndrome or are protective and clear Borrelia infection is the key to developing the next generation of both diagnostic tests and therapeutics to guide clinical management of patients being seen in the clinic. Next slide. And so this slide just highlights a couple vignettes of ways that we're doing this. So in this panel here, we're showing a very striking result from Lisa Blum in my lab, showing that when you compare people who return to health that are in the teal versus people who got post-treatment Lyme disease syndrome in the kind of the pink-orange, these are samples from the SLICE cohort provided by John Aucott and Mark Soloski. Basically, people who returned to health had a very high active B cell response, in contrast to people who develop post-treatment Lyme disease syndrome. And this suggests that there's just a dysregulation of the B cell response and an insufficient B cell response that is responsible for the post-Lyme treatment disease syndrome as compared to individuals who return to health. Down here, you can see another result that we found with John Aucott and Mark Soloski, basically showing elevated levels of a chemokine, CCL19, in individuals at the earliest times of infection that then progress to have post-treatment Lyme syndrome symptoms. And so CCL19, the cytokine, was higher in those individuals that develop persistent symptoms, and it suggests that there may be an inflammatory immune basis for that and provides insights into how you might further define the mechanisms underlying it, how you might diagnose individuals that will develop that, and how you might effectively treat it. And down here is another figure from Lisa Blum, and this is the antibody repertoire of a chronic Lyme disease patient. And in this repertoire, Lisa's finding antibodies that target various Lyme disease proteins. And when we collaborate with Monica Embers and she tests those antibodies for their ability to basically inhibit and thus block the growth of Borrelia, she finds that several of these antibodies are highly effective at completely blocking the growth of Borrelia in tissue culture. In addition, and what Lisa found to our great surprise, is that also within her antibody repertoire, there's certain antibodies being produced that target self-antigens. So these would be considered autoantibodies. And so a major question is, are these antibodies contributing to the clinical syndrome of post-treatment Lyme disease syndrome? And so studies in our lab are ongoing to further define whether this is in fact the case. And so basically we're doing what we term antibody repertoire sequencing, as well as T-cell repertoire sequencing. And these are based on DNA cell barcoding technologies that our lab developed 7 or 8 years ago, combined with next-generation sequencing. And the antibody repertoire sequencing lets us digitally break down a human's antibody repertoire into the individual antibodies that each B cell is producing, and thus provide molecular tools as recombinant antibodies that we can then define which ones are protective, so they kill Borrelia, and which ones are pathogenic, meaning they might target an autoantigen and cause an autoimmune process that would manifest in post-treatment Lyme disease syndrome. Now, don't get me wrong, I think post-treatment Lyme disease syndrome is a heterogeneous condition, and that there's multiple different causes to different subsets of patients' conditions. And so I think that the autoimmune component, we're garnering evidence that that's present, but it's likely a subset and potentially a small subset of those patients who have a certain autoimmune phenomena. And then the T-cell repertoire sequencing that we're working on with Mark Davis's lab at Stanford is to sequence the functional T-cell repertoire, so to identify what types of T-cells. In our world, there's Th1 cells, Th2, Th17, and T regulatory cells, and they each mediate different functions. And so with Mark, Lisa is mapping the types of T-cells that are associated with clearance and the types of T-cells that are associated with chronic Lyme symptoms. And you would target those, or you would enhance the ones that are protective and target the ones that are pathogenic with various drugs, some of which are already available today. So with that, I'll stop. Thank you.
All right. Thank you all for listening, and thanks to the Cohen Foundation for all the support they've given us for this work. I thought what I would just share with my 2 slides was a couple of just thoughts about things I think we still need to understand. And I show you this particular slide because this is a patient that was seen by John where there's a very clear erythema migrans. Now I know that some people don't display such a nice target lesion like that, but nevertheless, this is where I think the rubber hits the road in the beginning of the disease. This is where the tick bite occurred. This is where the tick has injected its saliva. Okay? This is where the tick has, the saliva is carried into it, some Borrelia burgdorferi, and there are some initial interactions with the components, the epithelial immune components that are resident in that skin, and it's driving what is a significant reaction. John has a infrared camera. And so he has pointed them at many of his patients, and this is a representative image of it. And it's, it shows that this is a pretty hot site. There's a lot of stuff going on in here, and we don't understand the pathways that are going on. And not only is this what's going on here important and impacting, I think, downstream events, but you can see it's radiating out. That the information that's happening here is radiating out systemically, perhaps to draining lymph nodes, etc. Perhaps the bug is being passed around. So what are the immune pathways that are here? How do they vary from individual to individuals? How do they influence downstream symptoms? Does the kind of rash that occurs or the fact that you can have multiple rashes, disseminated lesions, also have an impact on this? And how do we study this into a non-invasive manner? It's not easy to do. we take 2-millimeter biopsies, and most of that goes to sort of dedicated molecular diagnostics. I'd like to take a bigger biopsy. John will not let that happen because he cares about his patients. He doesn't want to do harm, and I'm an old mouse immunologist, and so I say, well, take out their brains. But we can't do that either, right? So we have to figure out non-invasive ways in which to investigate what is really going on right at the surface of the patient at a very early stage, because I think that those events are going to inform us about downstream events as well. The other thing I just wanted to stand up and talk about is that we have, I think, and echoing Bill's comments, is that we don't have a good grasp of the complexity of the T-cell immune response that is driven by Borrelia. And it's this T-cell immune response that's having such an impact on qualitatively and quantitatively on the humoral immune response. So I see T cells as being driven by antigens, but the antigens that T cells see might be a little bit different. They are a little bit different than what B cells see. T cells see short peptides, and the peptides that are seen that are derived from Borrelia proteins are different from individual to individual to individual. And so, what are the peptides that are driving this? Can we get a level of sophistication of understanding of this that we have in many other infectious disease settings? And this is sort of a schematic of an approach to do that using dendritic cells, which we know are the professional antigen presenting cells that we think drive immune responses. Perhaps Borrelia likes to do something to them that make them non-functional. We can take these derived dendritic cells and we can incubate them with known protein antigens from Borrelia. We have them cloned. Many people in this room have cloned these proteins. We know their N-terminal and carboxy-terminal sequences. We can feed them to the dendritic cell. The dendritic cell turns into an activated dendritic cell, which then can stimulate T cells. But what we want to do is purify those blue molecules here, that have these peptides, and then elute them and isolate them and analyze them by mass spec. And this is really important basic information because it will help us understand precise molecular structures that are driving T-cell responses, drive some new diagnostic assays. These peptide epitopes might be helpful in vaccine development. And you know what? Some of these bacterial peptides may actually resemble, they may be mimics of, if you will, of some self-peptides, so may be relevant to some of the self-reactive immune responses that are driven. So thank you.
All right, well, I think that set the stage nicely. There's a lot of technologies as well as a lot of approaches up here. I guess I wanted to, you guys all talked about this balance between kind of really understanding the mechanisms and being able to tease those apart versus then how do we translate that into humans? So what is, I guess, how do we do that? How do we take these really fundamental understandings about what the specific molecules are and then translate that up the chain into humans?
I think the first key is really understanding molecularly the mechanism, because that provides the key insights to design the diagnostic or the therapeutic intervention. So I think it takes longer to rigorously study mechanism than to take kind of a rifle shot at a potential target that might provide a near-term result. And perhaps there's a role for both near-term, more rifle shot attempts as well as a more mechanistic, defining of disease and based on which subsequent therapeutics are developed. But I think it really needs to go hand in hand to increase and make success a much more likely possibility. And if you look at companies like, any big pharma or biotech company, that's what they're doing, is they're defining mechanisms such that their therapeutic or their diagnostic is much greater opportunity to really have an impact.
Look what we've learned about in cancer immunology. Basic mechanisms that drove the understanding that, yes, there are T cells that seem to be active in a tumor patient, but they're quiet, they're silent. And they identified molecules called checkpoint molecules that were specifically upregulated to actually turn the T cell off. And now there's some marvelous new therapeutics that are working for some people, not all, so there's a lot of mystery still to be unraveled, but they are providing a whole new wave of immunotherapeutic approaches to, and maybe turning cancer into just another chronic disease. And so, I think there's lessons to be learned from them about the investment they've made in basic mechanisms and then outcomes in therapeutics.
I think it's extremely important to understand mechanism also from this. If you just think about when we started out, we were told that Borrelia evades immunity. There's no immune response, or there's no effective immune response because Borrelia hides in the skin. Well, we found 10 years ago now, so that that the first thing Borrelia does, it goes to the draining lymph node, the lymph node at the site of infection, which is the master tissue where all immune responses are induced. Rather than hiding and getting away from the immune response, that data showed that, in fact, the Borrelia is messing with the immune response, it's affecting how the immune response is being induced. That is profound because it changes how you think about how you want to treat it. Instead of making the Borrelia visible, you're realizing that it's already visible to the immune system, but not in a good way. And so clearly the therapeutic approach, whatever it may be at the end of the day, will have to be completely different. So you have to understand why before you can understand how you can treat it, in my view, for having long-term success.
So, just thinking with respect to antibiotic treatment, we've seen this difference in immune responses that seems to be driving patient outcomes. And it makes sense if you're treating your patients with doxycycline, which is a microbiostatic antibiotic, does not kill the Borrelia. It relies on the immune system to help clear the infection. So it makes sense that if patients are having different, we're seeing a different immune response in animal models, and now we're seeing the differential in humans. It makes sense that that type of treatment regimen would work for one patient but not for another. So when it comes to personalized medicine, what we learn from the basic science can be translated in that way.
And I was just going to take Mark's analogy further in terms of cancer. We don't just say cancer. Certain mechanisms work for squamous cell cancer, certain work for, and so I think on the clinical side, we have to define these subgroups of patients. They're not all chronic Lyme patients. There's subgroups. There's some people that may have an autoimmune bias, or some people that may have a persister bias. So it's defining those specific subgroups so that we can define the mechanisms in the subgroups. So it's that combination that gets to answers, the deep mechanisms, but in well-defined, narrow subgroups of patients.
So one thing Nicole brought up was this interesting idea of a coevolution, and you have a species or an organism that Borrelia persists in without apparent harm. Should we be attempting something like that? Like, how can we push? Should we start driving human evolution toward that, or what's the sort of similarities or differences between mouse and humans? Where what should we be doing along those lines?
I don't think you really want to propose this, so what you just said. So I think the easier treatment is that, and I think that's where the individualized medicine comes in, and the huge impact that this big data analysis that we talked about in the morning can have, and that rather than defining the mechanism, which I think would be incredibly difficult, but you can define the pattern of the individual and base the therapy on whatever the response is, because just as John said, not every patient is helped by giving them long antibiotics or even better antibiotics, because the reason for the disease symptoms are not a persistent infection. It might be an overshooting immune response. In other patients, it might be a problem of persistence. In other patients, it might be the activation of the immune system in the wrong way. So I think that's where this sort of big data comes in. So I think we can't retro-evolutionary adapt to this infection, but we can devise the therapeutic strategies that are individualized and take into account all this big data that we are trying to gather.
So from a diagnostic point of view, it's been mentioned a couple times, either non-invasive or sort of better tissue. What would you want? Assuming that you could get what you wanted, people gave you lymph nodes, they gave you some other tissue, or what would it be? What would you want for helping the diagnostic aim to actually get toward better defined patient subgroups?
So as a clinician, I look at it two ways. Again, it's the pathogen and the host response. So we would love to have a pathogen direct identification, and we've made progress with that in blood samples and skin biopsy samples. But the real holy grail is, if I get a nerves tissue sample, or I had a brain biopsy last week somebody had done looking for Lyme disease, we don't have very sensitive stains for the actual Borrelia in tissue specimens. But then the flip side, I learned as a rheumatologist, they define a lot of other diseases based on the immune response. So I'd settle for an immune response marker at this point too. A SED rate works great if you have polymyalgia. If you have polymyalgia, it's high. You give treatment, it comes down, and the person's responding. I'd settle for that right now for Lyme disease.
Okay. All right.
I think really you'd like to identify the antigen. In the end, that's what everybody's looking for, because even the best biomarker is a correlate. So having the actual antigen, and it is so difficult because Borrelia doesn't want to be in the blood. It's not where it likes to be. Even in the mice, I showed you this data, only 10% of mice that we know we are infected, and we can take other tissues, we know the bug is in there, only in 10% of the cases can we find it in the blood, although we can 100% find it in any other tissue we look for. So, the question really is, if you want to go for this sort of holy grail of actually finding the antigen, I think you have to realize that blood is not the right place to look. And so, what would be a tissue that's accessible, that's clinically possible that you can take? That's really the big question. So, if we could answer that, that would be great.
So, Monica, what's going on with the monkeys where they were not treated but responded quite robustly, the same as the treated ones? I thought
They were not treated.
I thought, well, one of your slides, you had this really robust antibody response in a lot of the monkeys, some of them treated, some of them not.
So I think it's just like humans. Some monkeys respond really well with their antibody responses, and some don't. So what we did was we profiled the antibody responses over time in monkeys to different Lyme antigens, different Borrelia antigens, and we showed that the kinetics of the antibody response was different depending on what antigen you were looking at. So we've been working to try to develop a diagnostic test that utilizes multiple antigens and can pick up the infection at different phases of disease. But then you run into this problem of seronegativity, and this is something that I've been talking to John a lot about, and now that I see your data, I've had some of John's patients, and I've tested them with our diagnostic test. I've put them on Western blots, completely negative, and they're classic Lyme cases. So what do we do about that? I don't really believe that they're seronegative. I think we're just not finding the right targets. So we need to take a broad approach to identify any Borrelia antigens that could be targeted by antibody responses to pick up that infection. Or find the antigen, as Nicole mentioned, but that's very hard. With humans, you've got your choice between blood and urine and possibly CSF. And that's about it, right?
I think it's so important to realize that Borrelia, when you grow it up in culture, which is where we can get the antigens from, either we make them synthetically or we grow it in culture, the antigens we have in there are not the antigens you have in a mammalian host. So we're relying on the antigens that are grown in some way that will never be what it is. So, you got to screen animal models, because I don't know how else you would do it, that they're infected with a particular bug. And that's the only way I think you will find the antigens. Now, Steve Bartlett has done this for 20 years. He's found some antigens that are very important, some of which are in your panel. And maybe there needs to be more done of that. Maybe it can be done the way that Bill just described, going from the other way, looking at the antibodies and then sort of finding them. But even if they're a large panel of antibodies, just talking to at the poster session, only a small subset of them were identified the antigen. So it's not simple, but I think it's an important effort to undertake to really identify the antigens that are most important. And finally, what I want to say is those that we find to be of diagnostic value are unlikely those that we find to be of therapeutic value. Because if Borrelia keeps expressing them, it means it doesn't care that we make an antibody response to it. If it doesn't care we make an antibody response to it, it's because it's not neutralizing. Okay, OSP-C, which is a fantastic antibody that if you have antibodies against OSP-C, you're protected from infection. It's as simple as that. I mean, Monica, you've shown it in vitro, in vivo, it works fantastically. But OSP-C is downregulated within 7 days of infection, right? I mean, Borrelia doesn't want to have it expressed. We make a piddly antibody response to it. So the therapeutic targets are different from the diagnostic targets, distinct, different. And so what is good for one would be bad for the other.
I think it's just extraordinary that there are these, a good quarter, isn't it, John, if I get my numbers wrong, of folks who don't make a sustained, strong antibody response that we can detect either using standard two-tier stuff or more sensitive, bead-based assays that Bill's done and Monica's done, that these are, from an immunological standpoint, you would say these are non-responders. And what does that mean? And what is the mechanism behind that? I think T-cell studies, because T-cells impact high affinity antibody responses, and they drive them. So I think that's going to be an important, piece of the puzzle that needs to be figured out.
So I wanted to open it up to the audience for questions if there's burning questions. It looks like actually there's a few, so maybe we can get mics to. Yeah.
Hello, I'm Rick Ostfeld from the Carey Institute. So part of the discussion has revolved around appropriate animal models for the human Lyme disease system. And there's some tension about mouse versus monkey models. And essentially that comes down to, as it's been expressed, that the mouse is co-evolved as a reservoir host and the monkey is not. The monkey better represents humans as a dead-end host. But of course, we're talking about the lab mouse, Mus musculus, the house mouse, and it is not known to play an important role in an enzootic cycle in nature, whereas the white-footed mouse, Peromyscus leucopus, is a reservoir host in nature, and it has an old world counterpart, Apodemus. And the difference between a reservoir host and a dead-end host is really a quantitative difference, not a qualitative one. I guess where my question is going is whether there are some missed opportunities when it comes to wildlife immunology, comparative immunology, where in nature, anywhere around here in a Lyme disease endemic zone, you have dozens of species of mammals and birds that are all exposed to Borrelia burgdorferi. They're all bitten by infected ticks. And they all have some non-zero transmission back out to uninfected ticks, but that varies from 90% in the white-footed mouse to 2 or 3% in a skunk or an opossum or a raccoon. So they're not completely refractory, they're just highly inefficient, yet none of those animals are known to show any clinical signs or symptoms or pathology. Perhaps because they haven't been studied carefully enough, maybe we've just missed it. But I wonder whether there are opportunities to use other models that are real-world victims of Borrelia infection, but are neither permissive to infection nor show pathology as a response to that infection. That would be actually the majority of mammals and possibly birds in these native systems.
So we have done some studies in Peromyscus and didn't find them to be significantly different to the strains of mice that we are using. And since they are the ones we have all the tools for, that's what we are studying. So I think it shows that the immune responses, while they are variations, I think overall the immune systems are not that profoundly different. And you know, I'm a veterinarian, so I've studied a lot of different species, and it's variation on a theme. So I think we shouldn't overestimate the species differences, but I think there are some. But I agree with you that maybe for some species that we haven't studied, because I'm not aware of anybody looking in skunks and things like that, that there might be pathology in some of those, and we just don't see them. They die, and you know, they're made out of Lyme disease, but we don't know it. So I wouldn't rule that out as a possibility.
Well, I don't perceive tension between the mouse and monkey model. We get along just fine, but I understand your point, and maybe analogy is, you know, like some animals have responses to multiple tick bites, anti-tick immunity, and some don't. For example, the guinea pig responds very vigorously to a tick bite, and mice don't. So what can we learn about from that animal compared to the mouse? And I don't know that there's really been anything in that area. I know that there's been some interest in looking deeper into Paramiscus to find out how it's a permissive, non-diseased host. But that's a good point.
I have a question. So when my patients get reinfected, and unfortunately I have a fair amount of folks that do, and obviously, you know, they are sometimes reinfected with a different strain and, you know, some co-infections and so forth, but it's quite detrimental. People do not do well. They do worse than they did even the first time that they got infected, usually, is what my data is starting to show. I was just wondering, in your experience with human and animal data, what's the pattern? Do you see any immune system adaptations with secondary reinfection, and what should we expect?
Nev, you know, currently we've not studied people that had second infections. We see them too clinically, but they're currently not part of the enrollment. Sort of using the building block approach, we're kind of trying to understand the immune response to the first infection and then be able to layer that on top of it. So I think my clinical experience is like yours, though, that that happens, and I wonder whether these people are primed to more of the kind of brisk, you know, memory inflammatory response, and so they suffer a lot more inflammatory symptoms because they've already been primed and their immune system has a memory, but we haven't studied it yet.
Nicole, it seems a little counterintuitive to me that, you know, there's a robust B-cell response, you know, very early during infection, but yet the absence of IgM is protective. Can you elaborate a little bit on your thoughts about that?
Yeah, what I showed you is just one slide. So what our data is that IgM is really important to get rid of bacteremia. That's the data I showed you. Otherwise, IgM is not very good for you. In fact, we think it's actually detrimental. We find ongoing IgM responses in our mice. So in contrast to a normal infection, normal is being defined in my lab as influenza, responses to influenza, which we study. IgM continues to be high and continues to be de novo produced. And so we have some evidence that that is actually detrimental for the IgG response in the host. So IgM is really good in reducing bacteremia, but that's the only good thing we can find about it.
And Nicole, yes. I'm interested to know if you've ever studied the disease in a cat, because cats don't seem to be affected by it.
Yeah, this is very interesting. There's actually a German, you know, in a socialized medicine system like in Europe, you can do these studies where people take 8,000 blood samples every year from a cross-section of the population. And they found that there was an increase in serum antibody titers to Lyme disease. overall in the population as the population ages. But the only correlation other than age that was found was being a pet owner, being an owner of cats, of all things. So there was the idea that maybe dog owners would have increased levels because they go out in the woods with their dog for a walk. You don't often take a cat for a walk, although some people do. But so what seems to be the case with cats is that the ticks actually really don't like feeding on cats. They don't, and so what they do is they come into the house with the cat, but then they jump off because they really don't want to be on the cat, so they'll find humans or dogs. And so I don't know whether the lack of the cat as a species in which Borrelia seem to be happy is because they don't get infected at the rate, or they get infected and then cleared. And so from that serological data I just outlined, it seems that it's really the former, that the cat is not often infected because the tick doesn't really like to feed on the cat.
So unfortunately, I know that there's a couple of other burning questions, but we have to stop the session. So can we please thank the panelists? And if you have any burning questions, maybe there's a minute or two to come harass them.